I'd like to extend a really warm welcome on behalf of the Hereditary Disease Foundation. I'm Leslie Thompson. I'm chair of the Scientific Advisory Board. And it's very odd to be here for the first time after so long of Anne doing this, but it's really a pleasure to be here. Anyway. It's my honor to introduce our keynote speaker tonight, Jeff Kelly from the Scripps Institute of Research in San Diego. And he is the Lita Annenberg Hazen Professor of Chemistry. And I'll tell you just a little bit about Jeff. It's really an honor to have him here as the keynote speaker. He first did his chemistry degree at State University of New York College. He got a PhD in organic chemistry from the University of North Carolina, and his postdoctoral training from the Rockefeller Institute. Kept going back and forth to New York. And he has been the recipient for the research I'm going to tell a little bit about from all sorts of areas. He was inducted into the National Academy of Sciences in 2023, which is one of the top honors you can get in science, and also the Breakthrough Prize in Life Sciences in 2022, another incredible honor in science, and American Academy of Arts and Sciences. He's a fellow of the Royal Society of Chemistry. He's on the National Academy of Inventors, and he's someone that has taken something from the very basic science, from protein structure and understanding that the clumps of proteins that can accumulate in the brain in neurodegenerative diseases or in tissues such as heart, as you'll hear, cause toxicity to cells. They make cells sick and they can make them die. But he took an approach where he understood that structure and then developed a drug. And it's FDA approved now for this type of cardiomyopathy and polyneuropathy. So it's something with it that very rarely does that happen, that you go from the bench to the true bedside, and this is now an FDA-approved drug. He started a number of companies along these lines. He had this first through FoldRx, and then this drug was acquired by Pfizer, and the company acquired by Pfizer, and then also proteostasis therapeutics. So it's really going to be great to hear this story from Jeff. Thank you very much, Jeff. Welcome.
Well, I'd like to welcome all of you this evening to the Hereditary Disease Foundation Gala. And I think it's appropriate that it's a gala because, in fact, we have a lot to celebrate. As some of you may know, over the past 3 years or so, there have been numerous drugs introduced and FDA approved that slow down the progression of neurodegenerative diseases. And I'm confident that in the next 5 or so years, we'll have a drug for Huntington's disease. What I thought I'd do tonight is to try to tell you about how these drugs came about and how they work, and then we can talk at the end about anything you would like to. So there are a lot of diseases where dementia is the final outcome. Of course, dementia refers to severity of symptoms that are significant enough to impede the quality of daily life. And the diseases here are listed in order of the number of patients that they affect. But of course, all of these diseases are incredibly important because they affect loved ones. So it's fitting to start by introducing the central dogma of molecular biology. These diseases are proteinopathies, so we want to know where proteins come from. So, of course, DNA is transcribed into messenger RNA, and then messenger RNA is translated into proteins. So there are drugs now that target messenger RNA, and there are a lot of drugs that target the proteins that misfold and aggregate in these disorders. So I like to think about proteins as being like a pearl necklace that's unlatched. There are 20 different color beads, and those beads have a modest affinity for one another dependent on their chemical properties. And as a consequence, proteins have the capacity to undergo a process known as protein folding that's generally spontaneous, although far from perfect, as you'll see. So the notion is that as a protein comes off the machine called the ribosome that makes it in a cell, it is mostly unfolded, and then owing to the amino acid sequence and the chemical nature of the side chains of the amino acids, the protein adopts pairings that confer it to be folded. Now, what's a general truth is that when a protein comes off the ribosome, most of it folds, but there's also misfolding processes and aggregation processes that are linked to pathology. And for complicated to fold proteins like Huntington, that can be 50% of what's made, quite a substantial amount. But when we're young, there are protective mechanisms to take care of that, as you'll see. Now, proteins are pretty big, and so we don't really represent them as beads on the string, but instead we make these so-called ribbon diagrams where we sort of trace the chain. And as you can see, the structures that proteins adopt are rather intricate. And this is a rather small protein, and they get much more complex as the size increases. So there's a lot of biology that's evolved in all organisms to control this competition between folding, misfolding, and aggregation. And as I mentioned, when we're young, our bodies and our cells are really good at dealing with the misfolded portion, either by refolding it or by degrading it, and analogously, the aggregated portion can be directed to degradation processes like the proteasome or the lysosome. So this side of the competition is largely eliminated owing to evolved biology, at least when we're younger. Now, what I want you to take home tonight is that these are diseases of protein shape changes or conformational changes. So you have a number of proteins that do normal, important physiologic functions, and upon misfolding or aggregation, they can adopt these different type of shapes, a prominent one being amyloid fibrils. So this is a structure that's now very well defined. It's a feature of virtually all neurodegenerative diseases. You can see that the individual strands of individual protein subunits line up and make these very large so-called cross-beta sheet structures. Now, if you put these structures on some kind of geographic length scale, and you assume that a normally folded protein would, say, be the size of Central Park, an amyloid fibril would span from the East Coast to the West Coast. So these are huge structures. Clearly, they're not good for cells and tissues. But I think there's still a lot of arguments in the field about whether amyloid fibrils per se are the main drivers of pathology. And we'll get back to that issue at the end. So major shape changes need to take place to go from the normally folded state to the cross beta sheet structures that are implicated as being a driver. But in fact, there are numerous non-native structures that are formed. And a lot of people think that these are more important in driving the pathology because they're soluble, they can diffuse to receptors, and it's well established in the scientific literature that these non-native structures can perform a number of abnormal functions, which over the course of decades can lead to the loss of tissues that don't easily regenerate, like the nervous system. So nerves, of course, are very long cells. There's a cell body, there's an axon, and as you know, an electrical signal has to travel down the axon, and then the electrical signal is translated from one neuron to another through synapses and chemical messengers. And perhaps the most important feature about the brain for the purposes of this evening's presentation is that neurons are highly interconnected. And it's possible by strategically losing either the synapse connections or the whole neuron itself, you can lead to pretty global functional changes that lead to memory loss and worse. Autonomic nervous system dysfunction, which compromises organ function, and so on. Now, as I mentioned, there's a lot of machinery in the cell to combat protein misfolding and aggregation. But about a third of the proteins are sent outside of the cell. And there's much less in the way of protective mechanisms outside the cell. And so when proteins get sent out, they typically have to be folded. But oftentimes they undergo a conformational change instead of properly refolding, which happens perhaps a million times in the lifespan of a long-lived protein. Instead, perhaps because of a mutation or some other environmental change, aggregation predominates and a disease ensues. So we came up with a really simple idea 25 years ago. The idea was simply to say, okay, if these folded proteins can't aggregate, could we fashion a ligand that bound to them, and so long as they allowed the protein to function, the presence of the ligand would be stabilizing and would prevent these aberrant shape changes that led to degeneration. Really simple idea, but could you pull it off? Of course, that's always the challenge, because making a liganded protein that's still functional is quite a challenge, as it turns out. So we decided to work on the transthyretin amyloid diseases. To be truthful, we had no idea at the time we started that these would become the third most prominent amyloid disease. And the reason is that the wild type protein that all of us have can aggregate in the elderly, causing autonomic nervous system dysfunction and a cardiomyopathy. And there are well over 1 million patients who have that disorder. So transthyretin is a protein that's structurally very well defined. And when it's in the circulatory system, such as the CSF in the brain or the blood in the periphery, it dissociates, misfolds, and aggregates, process I'll have a little bit more to say about in a minute. And this process of aggregation destroys organ systems like the gastrointestinal system and the heart, the autonomic, peripheral, and central nervous systems, as well as the eye. And now we're starting to appreciate that a lot of neurodegenerative diseases that were classically thought to be CNS-centric, in fact, destroy multiple organs and tissues. Now, what's important is that the cells and organs that are destroyed by this disease don't even make transthyretin. So it's an outside-in toxicity mechanism, presumably linked to aggregation. And it's one of the reasons we chose this disease, because we thought we could possibly understand that. There's 3 forms of transthyretin that circulate. The form that potentially can aggregate has no ligands associated with it. So transthyretin, as the name implies, is a transporter of holoretinol binding protein and thyroid hormone. But 50% of the protein that circulates in you and I can misfold and aggregate, leading to the maladies I talked about. So we spent a lot of time trying to figure out how this protein aggregates, because we thought if we knew that from basic research, we could do a better job fashioning a ligand to stabilize it. The key thing that we found is that in order for the protein to adopt these non-native structures, including assembled ones, the slowest step in all of this was dissociation of the tetrameric form of this protein. So basically, there are 4 identically folded subunits that come together, and I've just colored them differently so that you can easily see them. Many, many proteins in biology are oligomeric. One subunit folds and then several of them get together and perform complex functions. So this was a good day because the dissociation of the normally folded form is the slowest part of the whole process. And that bodes well for the notion that maybe you could stabilize the native form to prevent all the aberrant forms from assembling. Now, it turns out that there are only 127 amino acids in this protein, but there are more mutations in the human population that cause neurodegeneration than there are amino acids. And the reason there are so many mutations is that it's quite easy to screw up a protein. And the way that it gets screwed up, as you might imagine based on what I just told you, is that a single amino acid change can make the tetramer come apart faster. If that's the case, of course, aggregation is more efficient and the patient's at higher risk for neurodegeneration. Now, the big breakthrough came when Ted Foss, who was a graduate student, was doing a really simple experiment. And he wanted to know, okay, how does this protein come apart? There's lots of possibilities. And it turns out the bottom pathway is taken. This is the dimer-dimer interface in the protein that's the weakest link. That was highly relevant because that's a small molecule binding site that's unused in you and I, and that's another one. Those sites are used in lower animals to carry thyroid hormone. But in humans, there are a number of thyroid hormone carriers that are much better than this one. And so it's unliganded, basically. So the notion was that we could simply fashion a ligand to those unoccupied binding sites, stabilize that fraction of the protein that was circulating that would potentially misfold, and prevent the whole process of misfolding and aggregation from occurring from newly synthesized protein. So we argued that this was a very conservative approach for ameliorating the disease because you didn't have to know which non-native structure was the pathology driver. And this is a problem that I think will haunt us for another decade before we figure all this out. But there's a big unless here, right? And the unless is these patients are symptomatic. They have a lot of amyloid deposited in the extracellular space. And everybody at the time, frankly, was betting against us, not because they were mean people, but because amyloid was the common feature of all these diseases. So why does this chemist think he can stop disease progression by inhibiting the aggregation of newly synthesized proteins? So that was the risk. But fortunately, we were able to ultimately raise enough capital to do the clinical trial. Let me tell you how we got there. So these compounds are called kinetic stabilizers. So basically what happens is that if you can fashion a small molecule that binds to the tetramer exclusively instead of the dissociative transition state, which these proteins have to go through in order to aggregate, then depending on how tightly the small molecule binds, you can increase this barrier proportionally. So much so that it doesn't happen when the drug is bound if you have a potent enough binder. And this turned out to be, in fact, validated in biological samples. So I don't want to go through this in detail other than to say structural biology was the basis for fashioning a drug that bound really carefully to this protein. And Evan Powers is really the star. He conceived of the ultimate solution to this, a drug called tafamidis. And even though structure-based drug design in principle gets you to the finish line faster, we still had to make a lot of small molecules and do trial and error experiments before we came up with a drug candidate. Now, one of the hardest things that we had to solve was the question of, okay, what concentration of the stabilizer do you need in human plasma to stop the aggregation process? And a Nobel laureate that was here in New York was diagnosed with cardiomyopathy just at the point we got permission to do clinical studies. And he was incredibly enthusiastic about helping us decide what the dose should be using him as the subject. And we took advantage of that. So the way we did this is that no matter what the genotype of the patient, so they could be wild type only or have a mixture of wild type and mutant protein, we to their blood plasma, we just added what we call a reporter tetramer. And the notion is that if we have enough drug present, we won't form these mixed tetramers because in order to form them, the tetramer has to dissociate. So subunit exchange is rate limited by tetramer dissociation, just like aggregation is. And so every 6 weeks, we did a dose escalation in this patient, and we found that 60 milligrams once a day was the perfect amount to stop tetramer dissociation. And that turns out to be the dose that everybody's on today. Now, the first clinical trial was done with 20 milligrams because somebody at the FDA decided that we were going to turn an amyloid disease into a lysosomal storage disease by making the protein undegradable, which frankly was illogical, but you can't really argue with the boss, and FDA is the boss when you're trying to get a drug approved. So as you see, the structure of the drug is very simple chemically, but it binds very strongly. This means that at 2 nanomolar, 50% is dissociated, but at 10 micromolar, which is the drug concentration on average, basically 99.9% of the binding sites are saturated. So we did a clinical trial. This was pretty risky too, because we used the neuropathic impairment score. And this score was used for doing clinical trials in diabetic polyneuropathy. There have been, Steve, I don't know how many trials, but there must be a dozen. Every single one of them failed using this scale. So you could imagine my trepidation. But it was deemed to be a good scale because polyneuropathy associated with transthyretin aggregation was argued to be a faster progressing disease. And indeed, after unblinding, this 18-month placebo-controlled trial showed that individuals treated with tafamidis versus placebo had a slower disease progression. And so based on this, the drug was approved in virtually every country except the United States. And the FDA wanted another trial for reasons that we can go into at the end, but it was almost as irrational as suggesting that we are creating a lysosomal storage disease. But they did finally approve the drug, as you'll see. Now, this is perhaps among the more important slides. So in a clinical trial, you always want the treatment group and the placebo group to be as closely matched in every attribute you can think of. Disease status, age, and so on. And these groups were very well matched. Turns out that 70% of the population that started out on tafamidis were responders. If you delay treatment by 18 months by being in the placebo group before you roll on the drug, now only 50% of the population benefits. This has turned out to be universally true in every single neurodegenerative disease trial since. So diagnosing these diseases early and getting the patients on treatment early is absolutely critical to get a good outcome. And of course, you're often left with the problem that nobody wants to develop a diagnostic or a biomarker until you have a drug. But, hopefully the world will eventually change and people will do this in parallel. Now, we didn't initially understand that the transthyretin amyloid diseases had dementia as a major component. And that's because all the patients died historically before they developed dementia. So the way this works is that the protein concentration in the periphery is 25 times higher than it is in the brain. And aggregation, of course, the speed of it depends on the concentration of the protein. So the brain pathology develops slower because the concentration's lower. So once we started fixing the problems in the periphery with not so great methods like liver transplantation, we found out that those patients had a slower progression of peripheral and autonomic neuropathy, but all of a sudden they started developing dementia about a decade later. And Yoshi Sikajima was the brains behind this, as well as Teresa Cleo. And moreover, Ann Young can tell you this is a big problem. These people generally develop focal neurologic episodes. So they're driving their car, and all of a sudden they lose consciousness from anywhere from a few seconds to longer. So it really compromises the quality of life. Okay, fortunately, and this wasn't designed either because we didn't know this was dementia till we were halfway through this drug development process. Tafamidis gets in the brain, not great, but probably well enough to prevent dementia. And that's actually been borne out now because the first patient was treated 15 years ago. And we haven't seen any dementia even at the 20 milligram dose. And this is the exposure at the 20 milligram dose. Undoubtedly, at 60 milligrams, it's higher. It might not be linearly related. We don't know yet. So I mentioned cardiomyopathy. And I put this in because almost all these central nervous system diseases do have organ involvement. And of course, if I'm not mistaken, Anne, many of these people die from autonomic nervous system failures that ultimately compromise function of organs like the heart and so on. So Matt Maurer is a brilliant cardiologist at Columbia. And decades before everybody believed it, he said, look, there's millions of patients who have this. People are just missing the diagnosis. And he along with Pfizer, designed a really effective trial. And in these trials, you want the p-value to be less than 0.5. So this was a really exceptional trial. And this is the basis on which the FDA approved the drug. And the curve doesn't look as good as it did in polyneuropathy because these patients are much sicker. But the number of patients was higher, and so the statistics look much better. Now, the way that anytime you see a clinical result like this, you should be optimistic. Based on what I told you, that is, if you can diagnose the patients earlier, then patients that are newly on drug will do much better. And that, in fact, is starting to be borne out in these diseases, in the transthyretin amyloid diseases. And the story is the same here again about early treatment. So if you're New York class 1 heart failure, that is, you have mild pathology, taking tafamidis reduces the risk of death by 65%. If you progress to class 2, it goes down to 40%. So big change over the course of a few months even. Now, the reason to celebrate. There have been now, in the past 3 years, a number of new drugs that slow down neurodegeneration. The first is this antisense oligonucleotide. These lower the messenger RNA of the misfolding and aggregation-prone protein. So they drop the messenger RNA by as much as 80%. So the protein concentration goes way down. And this is a very challenging disease. Oops. This is superoxide dismutase-associated ALS. The trial failed, but they were smart enough to do an extension study. And what that means is after 28 weeks, there wasn't a big enough statistical distribution between the treatment group and the placebo group. But after everybody rolled on to drug, you see a nice separation between those individuals that were on drug the whole time versus half the time. And this drug has now been approved for ALS for good reason. This is a substantial disease modification. Now, this is a very particular form of ALS associated with the mutant form of superoxide dismutase. The other huge accomplishment, of course, is in Alzheimer's disease. I'm sure you've read a lot about this. The first antibody that came out wasn't that well accepted by the physicians. This is a second antibody that recruits microglial cells to the aggregates to degrade them. Now, this antibody also prevents soluble forms of A-beta from aggregating. And you see a pretty nice statistical distribution between the treatment group and the placebo group in terms of their disease progression. What you want, of course, is that these lines to be flat, no progression. But you need the patients to be sick enough in order to determine a difference. And so I think there's going to be this will get better and better as Alzheimer's patients get diagnosed earlier and earlier. And just last week, both Lilly, that developed another antibody that will likely be approved soon, and Eisai, who paid for these trials, both concluded that treating the patients early has a major effect on the extent of disease slowing of Alzheimer's disease, as you would expect based on what I've told you. So there are a lot of drugs now. I only told you about a few of them. There are half a dozen that treat transthyretin amyloidosis, either by stabilizing the protein or knocking down the messenger RNA. There are these two microglial recruiting drugs that slow down Alzheimer's disease. There's tofersen that slows down this genetic form of ALS. And just last week, there was a major report that in antisense oligonucleotide drugs that lower tau slow down the progression of Alzheimer's disease. That data hasn't been shared yet, but only a complete moron company would announce that and not have the data to support it. They would get eliminated. So I think the data will be strong. So I want to thank you for your kind attention. I hope I've conveyed a little bit about how these drugs work and why we should be optimistic. And I want to say that the Hereditary Disease Foundation is the one that's been in this Huntington's fight for a drug the longest. And I'm very confident, as I said at the outset, that we will have drugs for this disease in the next decade, certainly, and perhaps even in the next 5 years.